Okay, so starting with our nitrate reductase test, we are going to obtain a tube of nitrate broth and label it accordingly. We will then incubate it at 37 degrees Celsius for 12 to 48 hours. After the incubation period, we will inspect each tube for growth. If there is no growth, we will label no growth. And for the tubes that we do see growth, we will add in first 20 drops of the sulfonamic acid or nitrate A and mix well. And then after the nitrate A, we will add in 20 drops of the alpha naphthylamine or nitrate B and also mix well. Then we will let the broth stand for a few minutes and mixing it frequently so that the reagents can penetrate. And after a few minutes, if the broth turns red, then this is a positive reductase result. If we do not see change after a few minutes, then we will then add in a few particles of the zinc dust. And after we add in the zinc dust, we want to mix that well. And if there is a color change to red, then it is a negative reductase result. But if it again does not change, then that is a positive reductase result. All right, so for our citrate slant, we will obtain a tube that contains the citrate, and we will then take our inoculating needle and place it into the micro incinerator to sterilize it for 10 seconds. And after 10 seconds, we'll wait around a minute for it to cool down. And then once it is cooled, we want to take our inoculating needle and inoculate it into the citrate slant about 3/4 of the way down. And then we also want to streak the slant surface like that. Oh, and then I forgot to mention that before we inoculate it with the needle, we want to sterilize the lip of the tube in front of the micro incinerator for a few seconds. Okay, so after we sterilize the tube, we will then inoculate the citrate with the needle 3/4 of the way down, and then inoculate the slant, or we will streak the slant surface. And then we will take our inoculating needle, place it into the micro incinerator for 10 seconds to sterilize it. And then after that, we will incubate the inoculated tubes for 48 hours at 37 degrees Celsius. If there is no growth, then the slant will be returned to the incubator for an additional 72 hours so that we can confirm it is a negative result, because some species will take slower to grow on the citrate slant. And then after around 5 days, then we can be sure that the negative result is really negative. If there is growth and there is a color change from the green to the blue, then we can say that it's a positive result.